Hello, I am Yin Hoon. I am currently based at Mount Sinai School of Medicine in New York. Today, I am going to talk about a project that I have been working on in the CAR lab. And the title of my presentation is Towards a Human Whole Cell Model, a Prototype Model of Human Embryonic Stem Cells. My project has a long-term vision. We want to develop comprehensive models of single cells that integrate diverse data into a mechanistic understanding of every gene function. We envision that such models could help us personalize medical therapies to individual patients and also to help us engineer useful bioproducts. This vision was inspired by the whole cell model of Mycoplasma genitalium that was published in 2012. This model accounts for almost the whole genome. It integrates diverse data from more than 900 publications, and it mechanistically represents different cellular processes such as transcription and translation, as illustrated by the colored arrows in the diagram. These processes describe the interactions among different molecular types such as protein, RNAs, and metabolites. The success of this model inspired me to build a whole cell model of human cells. More specifically, I am prototyping a whole cell model of human embryonic stem cells. There were a number of reasons why we decided to focus on stem cells. First, stem cells have special ability to self-renew. Therefore, they form a well-defined clonal system, much like bacterial cell cultures. This property makes some of the approaches used to calibrate bacterial whole cell model applicable to stem cells. Second, stem cells are highly proliferative. Therefore, they have high growth rates and short doubling times, which are advantageous in terms of the computational time needed to simulate the whole cell cycle. Third, stem cells are karyotypically normal compared to cancer cells. And last but not least, stem cells have great potential for regenerative medicine. There are many commonly used stem cell lines in the literature. However, we have decided to model the H1 cell line because it is relatively well characterized and therefore has a lot of data that will be useful for modeling. However, there are many challenges to scale from modeling prokaryotic bacteria to modeling human cells. One of the biggest challenges is the genome size. There are only about 500 genes in Mycoplasma genitalium, while human cells have more than 20,000 genes. This means that to model human cells, we have more genes to represent, more reactions to code, and more data to handle. Besides that, human cells are eukaryotic cells, so there are additional complexities such as compartmentalization and alternative splicing. Computational demand is another challenge. When the model is bigger, there are more reactions to simulate, and the doubling times of human cells are also longer. To address some of these challenges, I have been working with my colleagues in the lab to develop a pipeline for building whole cell models. We designed this pipeline so that it will meet a few specifications. First, we want a pipeline that is scalable. That is, we want a pipeline that is able to build small bacterial whole cell models as well as large human whole cell models. We want a modular pipeline so that individual parts can be reused. Since we will be dealing with a large amount of data, we also want to be able to trace sources of data and be transparent about the assumptions and decisions made by modelers. This will allow us to make our models more reusable and reproducible and ultimately facilitate collaboration. This diagram here illustrates the architecture of our pipeline. The first step in our pipeline is to combine both qualitative and quantitative data into a knowledge base. The data in the knowledge base are then used to estimate the initial conditions of state variables and to generate pathway submodels. The initial conditions and the pathway submodels then form the whole cell model that we can simulate. In the next few slides, I will provide more details on each of these steps. To build a whole cell model of H1, I first needed to build a knowledge base. There were a few features necessary for building the H1 knowledge base. First, because H1 data was scattered across multiple sources, I needed software methods that would help me obtain this data easily. Second, the data were very diverse, so I needed a schema that would combine them. Finally, I wanted the resulting knowledge base to have an organized structure so that it would be easy to inspect, query, and analyze the data. This is how I built the H1 knowledge base. I manually curated data from individual papers and wrote custom code to parse the data. For data from public databases, I queried the data through Datanator that was developed by my colleagues that integrates multiple genomic and biochemical databases. I also created a knowledge base schema to represent the different types of data describing eukaryotic cells. The resulting knowledge base comes in two formats. The first format is a set of Python objects that are related through object relational mapping. Using an I/O tool developed by my colleagues, this set of Python objects can be written into an Excel file that is useful for storage and visual inspection. The Excel file can in turn be read back into Python objects for further analysis or model development. Currently, the H1 knowledge base consists of multiple omics, biochemical, and physiological data. This table shows the current content of the H1 knowledge base. I would like to mention that for genomic data, we are using the HG19 human reference genome, whereas for metabolic network, we are using Human Recon 2.2. Here, I show you a screenshot of the H1 knowledge base in Excel format. Each worksheet represents a different class of objects in the knowledge base schema. For example, I am showing a worksheet for the regulatory modules class, which combines data on promoter activity and type for each gene, and the transcription factors that regulate the gene. If the direction of regulation is known, as shown in this cell here, it will also be recorded. Once I had the H1 knowledge base, the next step was then to build a model from the data in the knowledge base. There were a few specifications to be considered in the design of this step. Since different pathway submodels contain different reaction patterns, I needed software methods that would automatically generate the reactions in each submodel. I also needed a single model schema to represent all submodels and the initial conditions of state variables. Another consideration was a storage format that would be easy to read and understand. This box here illustrates how I built the H1 model from the knowledge base. For each submodel, I developed a separate module in this Python package that automatically generates the reactions. The submodels generated by these modules are described using a language called WCLang, which is a model schema developed by my colleagues. WCLang can represent not only the reactions, but also rate equations, parameter values, and the initial conditions of state variables. Again, the resulting model comes in both Python objects and an Excel file. There are a few steps involved in generating each pathway submodel. The first step is to generate reactions based on genome annotation and network data. For metabolism submodel, we directly use the metabolic network in the knowledge base. However, for submodels such as transcription and translation, we use data in the genome annotation. Here, I show an example of an RNA degradation reaction. For each transcript in the genome annotation, we parse through the sequence FASTA file to determine the counts of each individual nucleotide. And these counts are then used as the stoichiometric coefficients in the reaction. Once we have generated all the reactions, the next step is then to generate a rate equation for each of the reactions. Because we have many different types of reactions, we follow some general guidelines to help us decide on which rate law to use. Generally, we include all known factors in the rate equations. These factors include enzymes, substrates, modifiers, and other types of regulatory elements. For reactions with one enzyme and one substrate, we use the Michaelis-Menten equation. And for reactions with multiple factors, we use a Michaelis-Menten-like term to represent each factor and then multiply all these terms in the rate equation. The final step is to calibrate the submodel to measured data. Due to a lack of single-cell measurements, we use species concentrations and cell growth measured over a population of individual cells. There are currently 7 submodels in the H1 whole cell model. They are transcription, RNA degradation, translation, and protein translocation into various compartments, protein degradation, complexation of protein subunits into enzymes, macromolecular formation of lipid and carbohydrate, and last but not least, metabolism. I am currently working on 2 versions of the H1 whole cell model. The first version is a full model that contains all the annotated genes and metabolic reactions. However, there are some challenges to working with this model. Due to the large size of this model version, it takes a long time to generate the full model. More importantly, simulating this model will be computationally expensive. Therefore, I have also developed a reduced version of the H1 model. The reduced version contains only about 700 genes. I only included genes that produce all the necessary RNAs and enzymes that catalyze the reactions in the submodels I have, and also the transcription factors that regulate those genes. For the metabolism submodel, I have also reduced the number of reactions. I did this by removing enzyme-catalyzed reactions that did not affect the simulated growth rate up to a certain tolerance level. Here, I show another screenshot, which is of the H1 model in Excel format. The worksheet I am showing is a list of all the reactions in the model. All these Excel files are stored in our GitHub repository, and we will make them public. To simulate the model, we are using a multi-algorithmic approach where we use different simulation algorithms for different submodels. Specifically, we use the stochastic simulation algorithm to simulate submodels such as transcription and translation, that contain reactions that are relatively slow. For metabolism submodel, which contains reactions that are relatively fast, we use dynamic flux balance analysis. This method is commonly used for simulating genome-scale metabolic models where there is a lack of kinetic measurements. I am using a multi-algorithmic simulator called WCSim that my colleague developed. In WCSim, simulations from different algorithms are numerically resolved through a set of shared state variables made up of species populations. The FBA is run at a fixed time interval, where at each time step, the simulator will read the populations of enzymes to constrain the metabolism submodel. The simulator will solve for the maximal flux through a biomass reaction, and this flux is used to update the populations of metabolites. On the other hand, SSA is run at exponentially distributed time intervals. All the reactions in submodels simulated using SSA are combined and evaluated together. Immediately after a reaction is selected to fire, the state variables will update the change in populations, and the updated values are used to re-evaluate reaction rates and to determine the next time step and the next reaction to fire. I have begun to run some trial simulations of my reduced model, and I look forward to sharing simulation results in the near future. To summarize my presentation, we have built a scalable pipeline to develop whole cell models. All our software packages are available on GitHub. I have described how I built an H1 knowledge base and used the knowledge base to generate both a full and a reduced whole cell model. I am currently running some trial simulations of the reduced model, and we aim to use the model to investigate the effects of stochasticity on cell heterogeneity. I would like to end by acknowledging our funding agencies and my colleagues in the lab, particularly Arthur, who developed WCLang and WCSim, Yusuf and Yang, who developed Datanator, and Yasmin, who worked briefly with me on the knowledge base. Thank you.